Hi there, I'm Maitreya Dunham, a professor at the University of Washington Yeast Resource Center. In my lab, we're interested in studying genome evolution. One of the tools we use to study this process is experimental evolution, where we evolve cultures of yeast in the laboratory and then ask how their genomes change. One of the devices we use to study this is the chemostat. The chemostat is essentially a culture vessel in which you can grow yeast cultures for many hundreds of generations, months in the lab, and then ask how the cells adapted. This chemostat here is a custom glass-blown chemostat, and as you might imagine, these are expensive and take up a lot of space. So recently in my lab, we developed the mini chemostat. The mini chemostat lets us do all of the things with the large volume chemostats, but for less money and in less space. My graduate student, Aaron Miller, is going to take you on a tour of how to build and use the mini chemostat array.
Hey everyone, I'm Aaron Miller, a graduate student in the Dunham lab. In this video series, I will show you how to make and use an array of ministats. If you have any questions that are not answered in these videos, please refer to the ministat manual. The ministat chamber is made up of a 50 ml glass tube stopped with a silicone cork. Air is delivered to the chamber by way of a long spinal tap needle, which reaches to the bottom of the tube and delivers robust airflow. Sampling is controlled by a second needle, which routes the culture to a collection chamber for analysis. The length of this needle determines the working culture volume. A third and far shorter needle is used to add media to the culture chamber. This activity determines the dilution rate and directly controls the rate of cell growth. The 20 ml culture volume stands out as being small enough that it can be greatly multiplexed and still fit in a relatively